Let's make meiosis a little less scary and a lot more interesting. Today, we're talking about meiosis 1, the stage where chromosomes basically meet up, mix things around, and then go their separate ways. First up is prophase 1. This is where homologous chromosomes, one from mom and one from dad, pair up like dance partners. They even swap pieces of DNA in a process called crossing over. Think of it like chromosomes trading recipe cards to create unique combinations. This is one reason why every person is genetically unique.
1. Prophase 1 commences with a diploid cell. Its chromatin contains two sets of chromosomes, one inherited from each parent. During this phase, the DNA in the chromatin replicates and condenses into recognizable X-shaped chromosomes and its identical sister chromatids of each chromosome. The chromosomes couple up with their corresponding counterparts in a fascinating process called synapsis, generating a tetrad made up of four sister chromatids. The true magic now begins with crossing over. Segments of alleles swap across chromatids during this genetic exchange event, creating brand new gene combinations. As meiosis 1 progresses, the nuclear membrane vanishes, and spindle fibers emerge, guiding the movement of chromosomes.
Next is metaphase 1. The chromosome pairs line up right in the center of the cell, almost like they're posing for a group photo before splitting up. Spindle fibers attach and get ready to pull them apart. Then comes anaphase 1, the dramatic breakup stage. The homologous chromosomes separate and move to opposite sides of the cell. But notice this, the sister chromatids are still attached. They're not leaving each other just yet.
Metaphase 1, the moment of alignment, where homologous chromosomes line up at the cell's equator. Then comes anaphase 1, and the spindle fibers diligently separate the homologous chromosomes in each tetrad, pulling them to opposite poles of the cell.
Finally, we reach telophase 1 and cytokinesis. The cell divides into two new cells. Each cell now has half the number of chromosomes, which is super important for making sperm and egg cells later on. And that's meiosis 1. Mix, match, separate, and divide.
Telophase 1, one chromosome from each homologous pair takes position at separate poles, although they still remain in the form of sister chromatids. However, each chromosome is still composed of sister chromatids. Remember that the sister chromatids of each chromosome are no longer identical due to allelic exchange and crossovers. The spindle fibers then disappear and the nuclear envelope reforms around the chromosomes. Finally, cytokinesis occurs. Meiosis 1 ends with two genetically distinct haploid daughter cells.
All right, now we move into meiosis 2, basically the final separation stage. If meiosis 1 was the chromosome breakup, meiosis 2 is where the sister chromatids finally decide to go their separate ways. First is prophase 2. The chromosomes condense again, the nuclear envelope disappears if it had reformed, and spindle fibers start setting up for round 2. Think of it as the cell getting back into work mode. Next comes metaphase 2. This time, the chromosomes line up individually in the center of each cell, almost like they're standing in a single file line waiting for their turn. Spindle fibers attach to each chromosome from opposite sides. Then we hit anaphase 2, the main event. The sister chromatids finally separate at the centromere and move toward opposite poles of the cell. Yep, the chromosome twins are officially splitting up now. We reach telophase 2 and cytokinesis. Nuclear membranes reform, the cells divide, and we end up with 4 genetically unique haploid cells. These cells are now ready to become sperm or egg cells. And that's meiosis 2. Separate, divide, and create genetic diversity. Biology really said, let's make everyone unique.
However, unlike meiosis 1, DNA is not replicated until meiosis 2 begins. Also in prophase 2, the nuclear envelope disappears and spindle fibers fan out from the two sets of paired centrioles. During metaphase 2, the chromosomes in each cell align at the equator and attach to spindle fibers at either pole. During anaphase 2, the sister chromatids of each chromosome separate and migrate to opposite poles. Once the sister chromatids separate, they are called chromosomes. Finally, in telophase 2, the spindle fibers disappear, the nuclear membrane regenerates, and both cells undergo cytokinesis. Meiosis II ends with 4 genetically distinct haploid daughter cells, each containing only one set of chromosomes.